Hi everyone, my name is Sung-Yon Kim. I'm a professor at Seoul National University in Seoul, South Korea. It's a great pleasure for me to participate in this SSIB 2021 virtual symposium on the gutsy brain. My lab studies innate survival behaviors such as ingestion, thermoregulation, and social behavior, and how the brain is able to sense specific stimuli that triggers or moderates these behaviors from both within and outside the body, and how the brain integrates and transforms the multiple streams of information to appropriate behavior outputs, such as eating, turning on a heater, or attack. The topic I would like to cover today is our latest work on ingestion, specifically about the termination of ingestive behaviors by mechanical distension of the digestive tract. So, like the title says, the overarching question of my talk is really how it is that the ingestive behaviors, eating and drinking, come to an end. This basic phenomenon has fascinated physiologists since mid-19th century, which led to the idea that animals use negative sensory feedback signals arising from the digestive tract organs to monitor and limit ingestive behaviors. At the front line of such feedback signals are those generated from the oral and oropharyngeal cavities. In this one of the early experiments, Roland Bellows created an opening or fistula in the esophagus, such that any ingested food or water drains from the animal before it can pass further. He observed that a thirsty dog would engage in sham drinking for a few tens of minutes, then pause the drinking and then start drinking again. So this and later findings led to the idea that oral and oropharyngeal signals can temporarily satiate hunger and thirst. But the sensory origin of these signals, their pathways, and their brain mechanisms are poorly understood. And mechanosensation may contribute to this temporary satiation because chewing odorless, tasteless, and nutritionless gums has been reported to reduce appetite in humans. But again, their mechanism has not been explored. Other feedback signals arising from the stomach and intestines have been pretty extensively investigated. So in these classic studies, an American physiologist, Neil Miller, showed that inflating the balloon surgically implanted inside the stomach suppresses appetite for food. And Eugene Tobin similarly showed that gastric balloon inflation reduces appetite for water. And later investigations, such as those by Phillips and Poley, who used pylar cuff and performed gastric loading experiments, revealed that the degree of appetite suppression was dependent on the volume of distension, but independent from nutrition content, osmolality, or pH of the gastric load. So far, the vast majority of the literature has consistently pointed to a critical role for gastric distension or mechanosensation of gastric wall distension in the control of ingestion. And this has also been clinically utilized in the gastric balloon therapy for treating obesity, which was FDA approved in 2015. The inflated balloon inside the patient's stomach supposedly induces an early feeling of satiety during a meal, thereby helping patients to lose weight. Feedback signals originating from the intestine also control ingestive behaviors. And contrary to the mechanosensory nature of the signals from the stomach, intestinal feedback signals are primarily thought to arise from the sensation of nutrients. This notion is supported by studies showing that intraduodenal infusion of nutrient solutions like glucose, amino acids, or fat, but not the injection of the same volume of isotonic saline or water, reduces food intake. For example, this study has fairly clearly shown that intraduodenal infusion of fats robustly suppresses feeding, but the injection of saline does not have an effect. And further works by Jerry Smith, Terry Poley, and many others have convincingly established this notion that gastric satiation is volumetric and intestinal satiation is nutritive. So among the diverse kinds of feedback signals, how nutrients are detected in the intestines signal to the brain and regulates appetite has been a topic of intense interest. And in contrast, its counterpart in mechanosensation has received much less attention despite its established key importance in the control of ingestion and its clinical implications. Several notable recent studies coming from the labs of Zach Knight and Steve Lieberless have found that vagal GLP-1R positive neurons form igels in the stomach wall, which represent a candidate mechanoreceptor ending type. And about a quarter of these neurons are activated upon gastric distension, and activating this population suppresses both feeding and drinking. So these important findings establish vagal GLP-1R positive neurons as a peripheral mechanoreceptor that can transmit appetite-suppressing signals in response to gastric mechanosensation. However, a specific population of neurons in the brain that encodes mechanosensation in the digestive tract is yet to be identified. And the circuit mechanism by which negative mechanosensory feedback signaling from the digestive tract reduces appetite remains largely unclear. And there is still no evidence showing the requirement of vagal GLP-1R positive neurons in gastric distension-induced appetite suppression. So there could be multiple other pathways contributing to this phenomenon. So we set out to address these questions. But where should we start? The digestive tract, the oral and oropharyngeal cavities, the esophagus, the stomach, and intestines. The walls of these organs are extensively innervated by cranial nerves 5, 7, 9, and 10, and also by spinal afferents. These nerves transmit sensory information to the nucleus of the solitary tract, or NTS, in a visceral topic labeled-like manner, and also to the trigeminal nucleus in the caudal hindbrain. And both of these nuclei strongly project to the parabrachial nucleus, which is a small pontine structure that broadly innervates many forebrain and midbrain areas that are implicated in feeding and drinking. So this architecture makes the PB well suited to play a key role in mechanosensory monitoring and feedback control of ingestion. So we focused on the PB, and among the heterogeneous subregions and cell types within the PB, we sought to identify a genetic marker for the specific PB subpopulation that is first naturally activated upon ingestion, and that encodes mechanosensory signals from the digestive tract, and third, that transmits appetite-suppressing signals. Towards this goal, we first examined the parabrachial nucleus that are activated simply upon water ingestion. And specifically, we analyzed the expression of FOS, which is a surrogate marker of neuroactivation, after providing free access, water access to thirsty mice. And what we observed is that FOS-positive neurons were highly concentrated in the dorsal lateral subnucleus in a remarkably localized manner throughout the anterior-posterior axis of the PB. And from the Allen Brain Atlas, we found that this particular subregion of the PB is marked by the expression of the prodynorphin gene. So this led us to hypothesize that PB prodynorphin neurons might be selectively activated upon ingestive behaviors. To test this hypothesis, we repeated the same FOS experiment with a fluorescent PB prodynorphin reporter mice. And we found that most of the drinking-induced FOS-positive neurons were prodynorphin positive, and most prodynorphin positive neurons were also FOS positive. So these data indicate that prodynorphin might be a useful genetic handle to target the PB subpopulation that is activated upon ingestion. We also did some fish experiments to show that the prodynorphin positive neurons are glutamatergic and are completely separate from KLK-positive CGRP neurons and partially overlap with oxytocin receptor neurons, about 20%. We then assessed the real-time activity of these neurons using fibrophotometry. As many of you are familiar with, fibrophotometry is a way of recording bulk calcium activity from a defined neural population expressing GCAMs. As you can see, PB prodynorphin neurons are acutely activated upon water intake, and the robust responses are highly time-locked to individual drinking bouts. But are these neurons responsive just to water? It turns out that these neurons could also be activated by the intake of diverse liquids or fluids, including basic bicarbonate solution and liquid food and hypertonic saline, as well as silicone oil. And these neurons were also significantly activated upon the consumption of hydrogel and chow food. And to explain for the difference in response amplitudes, we plotted the average response amplitude against intake rate and found a strong positive correlation. So these data suggested to us that PB prodynorphin neurons can monitor the ingestion of any types of solids and liquids using mechanosensory signals from the digestive tract. So why not try out directly applying mechanical stimulations? We found that PB prodynorphin neurons were strongly activated by touching the tongue, and these neurons were also robustly activated by inserting a gavage needle into the esophagus. And these neurons were strongly responded to gastric balloon inflation as well. We also noted that maintaining balloon inflation for an extended duration revealed an adaptation-like responses. So PB prodynorphin neurons seem to encode instantaneous changes in the volume of the digestive tract, rather than the steady state extended volume. By contrast, PB prodynorphin neurons did not respond to the distension of the duodenum, nor to the distension of the proximal colon. So the activity of PB prodynorphin neurons appeared to encode mechanosensation from the upper digestive tract parts, but not the lower digestive tract parts. And apparently, these results align well with the previous notion that gastric satiation is volumetric and intestinal satiation is nutritive. We further conducted a variety of control experiments. First, the responses of PB prodynorphin neurons were not modulated by the taste, osmolality, or temperature of ingesta. So these neurons likely do not receive gustatory, osmosensory, or thermosensory information. And second, these neurons were unresponsive to the tactile stimulation applied to the dorsum or whisker. These neurons are not responsive to the sensory detection of palatable food as well, nor to the condition tone predicting water delivery. The activity of these neurons were also unrelated with locomotor activity or the anxiety level. And neither the baseline activity nor the responses of PB prodynorphin neurons were modulated by the third state. So these neurons respond to water intake to the similar extent, regardless whether the mouse is thirsty or not. To summarize our data so far, using fibrophotometry, we have shown that the PB prodynorphin population is activated by seemingly very distinct stimuli, ranging from water and food intake to mechanical stimulation of digestive tract parts. But how are these stimuli represented by individual PB prodynorphin neurons? Are the same or different neurons respond to these stimuli? To address these questions, we turn to two-photon calcium imaging. And for the sake of time, I won't go too deeply into the two-photon datasets, but here's the data that are directly relevant to the question. First, we found that more than 80% of individual PB prodynorphin neurons were significantly activated by water intake. The majority of PB prodynorphin neurons robustly responded to gastric distension as well. And most of individual PB prodynorphin neurons also strongly responded to oral delivery of a liquid drug. A drop of water would not immediately reach the stomach. So here we are assuming that this is a stimulation applied just to the oral cavity. And strikingly, registration of the neurons across these three experiments revealed that most PB prodynorphin neurons responded to all three of these stimuli, namely water intake and gastric distension and oral stimulation. So from these experiments, we could draw two conclusions. First, most individual PB prodynorphin neurons represent integrated mechanosensory signals from distinct organs of the upper digestive tract, because most of the individual PB prodynorphin neurons responded to the stimulation applied to both the mouth and the stomach. So mechanosensory signals generated from distinct digestive tract organs, the mouth, the stomach, must have been integrated at the level of the PB, or maybe it's upstream and relayed to the PB. And second, these results show that individual PB prodynorphin neurons can monitor ingestion using mechanosensory signals from the digestive tract. And again, this is because most of the individual PB prodynorphin neurons were activated during natural ingestive behavior, as well as our direct mechanical stimulation. These data alone do not demonstrate that PB prodynorphin neurons or the brain monitor ingestion using mechanosensory signals, but it shows that it can do that rather than it does do that. Okay, so we next studied how the PB prodynorphin population receives mechanosensory signals from the digestive tract. To address these questions, we first mapped the monosynaptic inputs of PB prodynorphin neurons using engineered rabies viruses. The monosynaptic input neurons were found throughout the brain, but as we focused on the hindbrain regions to identify sensory input regions, we noted dense labeling in the trigeminal nerve nuclei and several specific subregions of the NTS. And as the audience of this conference would be familiar with, these are the specific regions that are known to receive sensory information from the mouth, pharynx, stomach by the cranial nerve afferents. And by allowing for additional synaptic spread of the rabies virus from the trigeminal nucleus or the rostral or caudal NTS, we could further label the anatomically connected neurons in the cranial nerve ganglia, including the trigeminal, geniculate, and nodose ganglia. And in addition to these cranial nerve pathways, we also found some spinal dorsal horn neurons are connected to PB prodynorphin neurons. So these results show that PB prodynorphin neurons are anatomically connected with both cranial and spinal afferent pathways. Now, as I mentioned before, the vagus nerve is well known to relay stomach distension signals to the brain. So would the vagus nerve be necessary for the encoding of stomach distension by PB prodynorphin neurons? Or do PB prodynorphin neurons receive stomach distension signals via spinal afferents? To address these questions, we tried vagotomy and spinal transection experiments, and the results were very clear. The responses of PB prodynorphin neurons to gastric air injection remained intact following spinal transection. And in contrast, these responses were fully abolished by subdiaphragmatic vagotomy. So mechanosensory signals from the stomach reach PB prodynorphin neurons via the vagal, but not spinal pathways. And this is consistent with the commonly held notion that physiological gastric distension is signaled by vagal afferents, whereas visceral pain signal is relayed by spinal pathways. So PB prodynorphin neurons monitor ingestion using integrated mechanosensory signals from the upper digestive tract, and the vagus nerve conveys stomach distension signals to PB prodynorphin neurons. Now, what do they do when they're activated? To probe the causal function of PB prodynorphin neurons, we used chemogenetic and optogenetic tools. First, chemogenetic stimulation of PB prodynorphin neurons potently suppressed food intake in food-deprived mice, and also suppressed water intake in water-deprived mice, as expected for the neurons signaling gastrointestinal distension. But the thing that surprised us was the observation that these effects were mediated by the reduction in the number of bouts, but not the duration of the bouts. And this is true for both feeding and drinking. So if the mouse became satiated for hunger or thirst during a bout, the duration of bout should have decreased because satiation would promote meal termination. But here, the bout duration was not affected, which indicates that activating PB prodynorphin neurons likely does not induce satiation. And optogenetic stimulation of PB prodynorphin neurons also had the same effects. So here we applied the laser stimulation as the mouse licked the spouts. So the stimulation was given only when the mouse already initiated drinking bouts. And this lick pair stimulation of these neurons again decreased the bout number, but not bout duration. So these results clearly demonstrate that activating PB prodynorphin neurons suppresses the initiation of intake bouts and reduces bout numbers, rather than terminating already initiated bouts and decreasing bout duration. Additional real-time place preference tests reveal that activating these neurons is avoided, and pre-stimulating PB prodynorphin neurons before the test session suppress water intake for at least 10 minutes, even if the laser is turned off. So we can infer that activating PB prodynorphin neurons produces a sustained appetite suppressing signals. So while the activation of PB prodynorphin neurons is strongest at the beginning of the intake, the appetite suppressing effects are exerted over the next tens of minutes, thereby decreasing the bout number. And it is important to note that such a decrease in bout number rather than bout duration is also observed when the stomach is distended. So this is an intragastric air infusion experiment that reduces gastric distension, that induces gastric distension without any optical or genetic manipulations. And here, gastric distension suppressed subsequent water intake by reducing bout number, but not bout duration. So our observation on the effect of activating PB prodynorphin neurons closely parallels the symptoms of gastric distension. And next, we chemogenetically inhibited PB prodynorphin neurons to assess their physiological roles. This manipulation increased food intake in food-deprived mice and also increased water intake in water-deprived mice, and elicited a trend towards increased intake of hypertonic saline in salt-depleted mice. What's evident from these results is that inhibiting PB prodynorphin neurons induces pathological overconsumption of a variety of food and drinks, but only for those that the mouse already has the appetite for. And the effect is reminiscent of the effects observed by the loss of function manipulations of many gut-borne biochemical signals. Together, our data demonstrate that PB prodynorphin neurons are required for the negative feedback control of ingestion. Then how does the PB prodynorphin population suppress drinking behavior? To get some clues to this question, we examine their projection targets. This is a 2 millimeter thick sagittal tissue section of the brain that expresses EOFP in the PB prodynorphin neurons. And you can see that the axon bundles of these neurons sort of bifurcate from its origin. The dorsal pathway goes to the PAG, and the ventral pathway goes through the midbrain areas, and then innervate pretty much all subregions of the hypothalamus, including the preoptic area, which then makes a U-turn at around the median preoptic area towards the BNST and SFO. As you're well aware, these are the many forebrain and midbrain areas that are critical for ingestive behavior. And I don't have the time to show this, but we also showed that the projection to the PBH can suppress feeding. So the PBH might be the key downstream target of the PB prodynorphin neurons. Okay, to briefly summarize today's talk, we have demonstrated a neural circuit mechanism centered around the hindbrain PB prodynorphin neurons for mechanosensory feedback control of ingestion. And upon the receipt of the mechanosensory signals from the digestive tract with the signals from the stomach reaching these neurons via the vagus nerve, PB prodynorphin neurons produce aversive and sustained appetite-suppressing signals that discourage the initiation of feeding and drinking. And the projection from these neurons to the PBH may play the key role in appetite suppression. Like any other scientific discoveries, our study raises numerous further questions. First, what are the downstream circuit mechanisms for appetite regulation? Where does the hunger, thirst, and GI distension signals integrated? And can we find the circuit-specific drug targets to mimic GI distension and also reduce appetite? And what are the specific NTS, trigeminal and cranial nerve ganglia neurons that signal digestive tract mechanosensation? And how does gut mechanosensation interact at and potentially are integrated with chemosensory, nutrient sensory modalities? And what is the anatomy and functional significance of oral mechanosensation? And finally, what are the actual ion channels, mechanically gated ion channels that transduce the mechanical stimulation applied to the digestive tract to the electrical neural signals? So these are some of the questions that we are currently following up on this project. And there's not much progress yet, but just to show you a glimpse of our current endeavors, we are currently using anterograde and retrograde viruses in combination with tissue clearing techniques to identify the peripheral mechanoreceptors that are connected to PB prodynorphin neurons and also their afferent pathways. And this movie shows the trigeminal ganglia neurons expressing EGFP pan-neuronally. And we can see the beautiful 3 divisions of this nerve, ophthalmic and maxillary and mandibular divisions. And it's interesting to see the lingual branch of the trigeminal nerve innervating the tongue. And this is a mid-sagittal section of the tongue. And we can see that the trigeminal axons in the fungiform papilla, circumambulate papilla, as well as in the filiform papilla, all of which may contribute to the mechanosensation and or its signaling. And we can also see the trigeminal axons in the hard and soft palates in these structures. So we are currently investigating whether these trigeminal sensory fibers are necessary for oral mechanosensation using the response of PV prodynorphin neurons as a readout. Okay, so with that, I would like to thank my students, especially Dongyun, Yuryang, and Minyu, who did a wonderful job leading the project that I talked about today. Also, thank my collaborators and funding. And one last advertisement, my graduate student Xiun is presenting our unpublished work on thermoregulatory behavior in this oral session. So I don't know if a replay will be available, but if it does, please check out our latest findings here. Okay, thank you so much for listening for a long time.